Welcome to this video on epithelia. This video will introduce foundational concepts for understanding the structures and functions of epithelia. I hope you enjoy it. Let's begin with an overview. Epithelia can broadly be placed into three categories: simple, stratified, and what I like to call the special epithelia. Simple epithelia are epithelia that are only one cell layer thick, hence the name simple. They are named according to their shapes and can be squamous, cuboidal, or columnar. Stratified epithelia, as their name implies, consist of two or more layers. They are named according to the shape of the cells on the most superficial aspect of the epithelia. So, for example, we have stratified squamous epithelium, stratified cuboidal epithelium, and stratified columnar epithelium. We will also talk about keratinization. Finally, we will touch on the specially named epithelia: pseudostratified, urinary, and respiratory. Let's start with simple epithelia. As already mentioned, they are only one cell layer thick, and this provides the first part of the name of each type. The second part of the name is given by the shape of the single layer of cells. So if, for example, the single layer of cells is squamous in shape, the epithelium is called a simple squamous epithelium. If the cells are cuboidal in shape, the epithelium is called a simple cuboidal epithelium. And if the cells are columnar in shape, the epithelium is called a simple columnar epithelium. Let's look at an example of how we assemble simple squamous epithelia into structures. Simple squamous epithelium is found lining all of the blood vessels of the body and is here given the special name of endothelium, as well as lymphatics. So you can imagine that we need to assemble these squamous cells into a circular structure, as shown in this schematic, with a lumen in the middle. This clearly demonstrates how a single layer of squamous cells can make up a blood vessel or a lymphatic vessel. Such a thin layer of cells is functionally important in these locations for easy diffusion of such things as oxygen or fluids across the epithelium. Now compare the schematic to a micrograph image of a lymphatic vessel, and you can clearly see the flattened basophilic nuclei and eosinophilic cytoplasm of the squamous cells surrounding a lumen. Let's now look at an example of how we assemble simple cuboidal epithelia into structures. Simple cuboidal epithelium is found in a number of different locations in the body, and one example is lining the tubules of the kidneys. Once again, you can imagine that we need to assemble each cuboidal cell into a circular structure, as shown here in this schematic, with a lumen in the middle. This clearly demonstrates how a single layer of cuboidal cells with basophilic nuclei and eosinophilic cytoplasms can make up a beautiful tubular structure. Simple cuboidal epithelia are located in places specialized for secretion, such as salivary glands, and in places specialized for diffusion, such as here in the kidney. Now compare the schematic to a micrograph image of kidney tubules, and you can clearly see the basophilic nuclei and eosinophilic cytoplasms of the cuboidal cells surrounding a lumen. Finally, for simple epithelium, let's take a look at an example of how we assemble simple columnar epithelia into structures. Simple columnar epithelium is found in a number of different locations, and one example is lining the gallbladder. Once again, you can imagine that we need to assemble each columnar cell into a structure as shown in this schematic. However, this time in the example of a gallbladder with a lumen on the outside. This example is not showing a circular structure like the previous slides, but is showing the absorptive columnar epithelial cells of the gallbladder projecting into the lumen of the organ. Simple columnar epithelium is a very good choice for locations such as the gallbladder, where absorption of water from the lumen helps concentrate the bile that is stored in the gallbladder between meals. To put this into context, here is a micrograph cross-section through a gallbladder. The lumen of the entire gallbladder is in the center, and there are projections of epithelial structures, like the one shown in the schematic, into the lumen of the organ. If we magnify one of these projections, then you can compare it to the schematic, and you can clearly see the basophilic nuclei and eosinophilic cytoplasm of the columnar cells projecting into a lumen. Returning to this overview, let's now look more closely at stratified epithelia. As previously mentioned, stratified epithelia are epithelia that consist of 2 or more layers. They are named according to the shape of the cells on the most superficial aspect of the epithelia. So there is stratified squamous epithelium, stratified cuboidal epithelium, and stratified columnar epithelium. We will also talk about keratinization. The naming convention for stratified epithelia starts with the very fact that they consist of more than one layer of cells, so they are stratified. The second part of the name comes from the shape of the cells on the superficial surface. So if, for example, the superficial layer of cells is squamous in shape, the epithelium is called a stratified squamous epithelium. If the superficial cells are cuboidal in shape, the epithelium is called a stratified cuboidal epithelium. And if the superficial cells are columnar in shape, the epithelium is called a stratified columnar epithelium. This is a rare epithelium, and we will not look at it in this overview video. Let's look at an example of a stratified squamous epithelium. Again, it's called stratified because it consists of more than one layer of cells. It's called squamous because the most superficial cells are flattened, and so together it's called stratified squamous epithelium. This is quite a physically tough epithelium, and so it is located in parts of the body that require protection against such things as abrasion or desiccation. Really nice examples of this type of epithelium are found on the palms of the hands and the soles of the feet. Let's look at an example of how we assemble a stratified squamous epithelium. As shown in the schematic on the right, there are multiple layers of cells, and this means the epithelium is stratified. The most superficial layer is squamous in shape, and so we now call it a stratified squamous epithelium. One additional term I haven't told you about yet, but I'll tell you now, is that thick skin is covered on its surface by a layer of keratin, and this adds the final part of the name of thick skin, which in its full form is stratified squamous keratinized epithelium. Keratin is an acellular layer that provides protection against physical damages and stresses. Now compare the schematic to a micrograph of thick skin. Again, there are multiple layers of cells, and so we call it stratified. Then, because the superficial layer is squamous, we add that to the name. And finally, we name the keratin as the final part of the full name of stratified squamous keratinized epithelium. Let's next look at an example of a stratified cuboidal epithelium. This is a fairly unusual type of epithelium, and the layers can be multiple shapes, but the most superficial layer is cuboidal, and so it is a stratified cuboidal epithelium. It can be found as a protective component of the ducts of glands, including mammary, salivary, and sweat glands. Again, looking first at a schematic, it is called stratified because it consists of more than one layer of cells. It's called cuboidal because the most superficial cells are cuboidal in shape, and so together it's called stratified cuboidal epithelium. Finally, compare the schematic to a micrograph of a duct from a sweat gland. There is more than one layer of cells, but the layer immediately adjacent to the lumen is cuboidal, so this is a stratified cuboidal epithelium. Let's now wrap up this epithelia video by looking at three special types: pseudostratified, urothelium, and respiratory epithelium. Pseudostratified can be a tricky epithelium. On light micrographs, and in this example schematic, it can appear stratified because the nuclei are at different levels in the cells. But when observed under the electron microscope, it can be confirmed that the cells are not stratified because they all come into contact with the basement membrane, and so this tissue is called pseudostratified. Respiratory epithelium is a pseudostratified epithelium. If we use conventional naming nomenclature, then its full name is a handful. It is a pseudostratified ciliated columnar epithelium with goblet cells. Phew! Fortunately, because this type of epithelium is exclusively located in the respiratory tract, we call it respiratory epithelium. It's a beautiful epithelium. Let's compare the schematic to the light micrograph. First, it looks stratified with more than one cell layer, but we know that it is not and that all cells rest on the basement membrane. Hence, it is a pseudostratified epithelium. In addition, this epithelium has two specializations that we have not mentioned until now. The first are goblet cells. They are called goblet cells because they resemble wine goblets. These cells are packed full of mucin granules and secrete mucus on the surface epithelium. Here in the airways, that mucus helps to clean debris out of the respiratory system. The second specialization is the surface cilia. Cilia work together with the goblet cells in the respiratory system to clear debris. The cilia are motile and sweep debris in one direction, away from the lungs. The final epithelium for this video is urothelium. It is found exclusively in the urinary tract and so is given its own name of urothelium or urinary epithelium. It is my favorite type of epithelium. As shown in this schematic, it is a stratified epithelium. What makes this epithelium unique are the dome-shaped surface cells. They look like umbrellas at their apical surfaces and indeed are often called umbrella cells. If you imagine the urinary bladder filling up with urine and pushing on the surface of these cells, you'll understand how, as the dome-shaped surfaces flatten out, it increases the surface area of the epithelium and thus the storage capacity of the bladder. If we compare the schematic to a micrograph from an empty urinary bladder, you can see these beautiful umbrella cells. This concludes this introductory video on epithelia. Thank you for watching. You may access the virtual slides used in this video by clicking on the UBC Virtual Histo link in the banner of this YouTube channel.